Okay, now we come to the important part, that is secondary immunity. I should have made one section slide. So this is a new section starting. Okay, the section on primary immunodeficiencies is covered. We covered some T cell disorders, some B cell disorders, some innate system disorders. Now we arrive at the secondary immunodeficiency. So this was one of your learning objectives. And what are the causes? Okay, what are the causes of primary immunodeficiencies you saw? What are the causes of secondary immunodeficiencies? Extremes of age simply. So premature infants are very prone to infection, developing infection. They have to be safeguarded in an ICU called the neonatal ICU or NICU, NICU. Okay. Elderly. So now we are saying that the elderly have to be protected against COVID because their immune systems are weak. Okay. Chronic severe malnutrition. So this is perhaps the most common cause globally of secondary immunodeficiency. That means weapons मिले थे तुमको बचपन से. It's not your primary immunodeficiency में weapon मिले ही नहीं थे. इसमें मिले थे but you lost them. How? Through severe malnutrition. Simply because you were not eating enough. Simply because your proteins were getting destroyed to meet your metabolic needs. You lost your weapons. You lost your immune system. So this is a very common cause of immunodeficiency in impoverished regions of the world. और neglected old people like that. So old people में ये सभी causes हो सकता है. We didn't write chronic disease, so वो भी याद रखो कि chronic diseases like hypertension, diabetes can also lead to immunodeficiency. Then exposure to environmental agents like drugs used in. So if you listen to the last lecture, I haven't posted it yet. I will post it soon. But if you went through the PPT, the diseases like rheumatoid arthritis, SLE, so they are autoimmune disorders. Okay, so autoimmune disorders, allergies, they need to be treated how? How do you treat them? Because the immune system is hyper functioning. आपको क्या करना है? Tolerance बढ़ाना है. So you need to improve the tolerance so that immune system is down regulated. So that you do by using some immunosuppressive drugs on occasions. So immunosuppressive drugs like for example hydroxychloroquine, which is being used in the COVID pandemic. What does it cause? Hydroxychloroquine. So it somehow protects against the cytokine release. Cytokine release that is happening after your TH cell activation or cytotoxic T cell. TH cell, not the cytotoxic one. The helper T cell interacting with the coronavirus antigen peptide complex is going to release some cytokine. So that cytokine storm can cause the severe acute respiratory syndrome, SARS, or the SARI, severe acute respiratory illness. So that you want to prevent. So you can use hydroxychloroquine. You can use IL-6, interleukin-6 monoclonal antibodies called tocilizumab. So that is also being researched in a trial. So I'm basically trying to say that immunosuppressive drugs, because we use them to suppress immunity for rheumatoid arthritis, for SLE. Classically, one important drug is infliximab used in rheumatoid arthritis, which is an interleukin-1 antagonist. Okay, I'm forgetting, but so you look up infliximab. So those are immunosuppressive drugs. Steroids or corticosteroids. Okay, what are these? Dexamethasone or betamethasone, cortisol. So they are in general anti-inflammatory, in general immunosuppressive. So we have them endogenously in our body to relieve the stress, but they also, decrease the inflammation. So they don't allow the inflammation to become more aggressive. And therapeutically also, steroids are given to suppress the immune system. But like I said, they are non-specific. So these are non-specific things. It's not like tolerance. Tolerance is very antigen specific. Tolerance through T regulatory cells, tolerance through clonal deletion. Steroids cause general immunosuppression. So they are used when you are giving, for example, a kidney transplant to a patient. So you are doing a kidney transplant, you don't want the graft to be rejected. You keep the patient on steroids. Okay. You also give them other immunosuppressive drugs like cyclosporin, tacrolimus. So all those things. Chemotherapy. So chemotherapy is the term used to describe all the cytotoxic drugs that are given in cancer. So it's the field of medical oncology. So any patient with a cancer, let's say a blood cancer or a solid cancer, will receive some chemotherapeutic drugs like cisplatin or paclitaxel, something. Okay, there are many drugs. Either they could be spindle formation, they could inhibit the mitotic spindle formation, or they could interfere with the DNA metabolism in some other way. Toxic DNA adducts it can form. So all those chemotherapeutic agents are meant to stop the rapidly dividing cancer cells, but they also affect all the other rapidly dividing cell populations That leads to your hair loss because hair follicles are rapidly dividing. It leads to your diarrhea and vomiting because your GI epithelium, your stomach lining hai, intestine lining hai, that is also rapidly dividing mucosa. So that is eroded. Chemotherapy can also affect your bone marrow. So when it affects your bone marrow, it leads to anemia, neutropenia. It leads to, so that is why when you give chemotherapy, before giving a chemotherapy, you have to check whether the cell counts are good enough. नहीं तो आप कीमोथेरेपी दोगे, पेशेंट का सेल काउंट इतना लो हो जाएगा कि उसको, they will develop pneumonia, they can die simply because of that. So if an overdose of chemotherapy is given, for example, there was a case where 10 times the dose was administered, okay, in one of the hospitals. So there was a inadvertently, when the doctor on call failed to see the dose and he accidentally administered a 10 times higher dose, the child died of, the child was recovering from an eye cancer, a retinoblastoma, but eventually he died because of an infection, because the cell counts dropped so drastically that it was very difficult to save him. They tried giving colony stimulating factors, GCSF, to stimulate the bone marrow again, but the child succumbed. Similarly, radiation therapy. So radiation therapy also causes damage to rapidly dividing cells. So radiation also affects your DNA, and it causes DNA damage, especially when the DNA is, whenever the DNA is unfolded, it is more vulnerable to damage. When it is folded in the form of chromatin, it is not going to be damaged easily. When it is unfolded at the time of division. So all the rapidly dividing cell populations are more vulnerable to chemotherapy, to radiation therapy. So radiation therapy also, as a result of cancer treatment or radiation, you can get accidentally. Like if you have seen the series on Hotstar, Amazon Prime, I think, called Chernobyl, it talks about the Chernobyl disaster, or when atomic bombs were dropped on Hiroshima or Nagasaki, or the recent Fukushima disaster, if you know, the Japanese earthquake that happened some years back. So if you receive that kind of radiation also, it can lead to immunodeficiency. And coming to the more important, the one which you all know, the infection. So many viral infections can cause transient bone marrow suppression, can lead to decreased cell counts and immunodeficiency. But the one major pandemic, you can say, because it's there everywhere, the major, at least it's an epidemic, if not a pandemic, because it's not so fast as COVID is happening right now. But the global epidemic is that of HIV/AIDS. And it's also important because the other viral infections generally cause transient bone marrow suppression and transient immunodeficiency. But HIV is a chronic disease and causes a long-standing ailment, finally resulting in death of the patient. The HIV epidemic. So in the early 1980s, there was suddenly an increase in a very selected, how do I say? It's a new kind of cases started appearing in the US, where along both coasts of the US. So along the East Coast, that is New York, along the West Coast, that is Los Angeles and San Francisco. No, Los Angeles or, yeah. So Los Angeles and San Francisco are on the East or on the West Coast. So on both these coasts, healthy young patients suddenly started presenting with pneumocystis carinii pneumonia and Kaposi's sarcoma. Kaposi's sarcoma is a malignancy found in skin. So these disorders normally were unheard of. They were only seen in patients of cancer getting, very high chemotherapy. So people didn't know, they hadn't heard of such illnesses. So this is the morbidity and mortality weekly report of that time. So patients presenting with, Kaposi's sarcoma or with pneumonia. This is pneumocystis carinii pneumonia or Kaposi's sarcoma. You can see the enlarged lymph nodes of a pneumonia case. So finally, there were reports published in New England Journal of Medicine. So this is the biggest journal of medicine, okay? Medicine or surgery or whatever you call it. Of the medical sciences, the most famous journal is the New England Journal of Medicine with the highest impact factor. So impact factor is one of the ways in which the importance, the value of a journal is judged. So this has an impact factor in 40s. Okay, this is like even higher than the basic science journals like Nature and Cell or Science. The New England Journal of Medicine, because it has medical applications. Generally, the medical journals have a higher impact factor than the basic science journal. Basic science journals the good ones include Nature, Science, and Cell. So original articles were published that pneumocystis carinii pneumonia and mucosal candidiasis in previously healthy homosexual men. Basically, the epidemiologists started studying that who were these cases, and they observed that Outbreak of community acquired pneumocystis carinii pneumonia. So see, community acquired, it has become community acquired. When we say here अभी COVID के केस में तुमने सुना होगा कि third stage of epidemic आएगा तो community acquired बन जाएगा. Community transmission has started. So when it becomes a community transmitted disease, that means you can't even trace the source. कि अगर आपसे मैं interview ले रहा हूँ, आपका पूछ रहा हूँ that where did you go in the last 1 week, who did you meet, even if you are telling all the answers correctly, still I am unable to find out where, because I cannot find any known positive case in your contact. That means that the community spread has happened and I don't know who all are the positives. So that is a community acquired pneumonia. So could be a manifestation of cellular immune dysfunction. So all these reports were published. Opportunistic infection. So this entire New England Journal was filled with all the cases related to cellular immunodeficiency, severe acquired immunodeficiency, and they found that it was majority were homosexual males. Okay, so that was the clustering. There is a very good movie on this, on the initial days of the HIV epidemic, how they started, because no one knew how to handle these cases. They were totally unaware of what was happening, how these cases were coming. किसी को ये थोड़ी लगा था कि ये एक डिजीज का पार्ट है, कोई एक अपोसिस सार्कोमा लेके आ रहा है, meaning some are coming with skin malignancy, some are coming with pneumonia. Only that these were rare illnesses, that's all. They only knew that these were rare illnesses. To make a network out of these, that they are all part of the same disease spectrum, took a long time. To understand that they were all being caused by a particular pathogen which is infecting the immune system itself. Okay, so it is not infecting a liver or your kidney or your brain. It is infecting your immune system, meaning it is that, like ताकत तो मेरे जितनी ले आओगे, but उतना what I don't know. Let's not go into my कुछ फिल्मी dialogue मुझे याद आ रहा था, but like उतना हरामी, I'm sorry, like उतना it is that evil that it is going to take the war to the war room directly. It is going to attack the immune system. It is, instead of attacking the cells, it is attacking the immune system. So they found, so that movie is known as And the Band Played On. ये movie का नाम है And the Band Played On. It stars Richard Gere. So they have discussed how they did the initial investigations. So on the initial investigation, they found that the clustering was happening in homosexual males. So this was the cluster that was primarily presenting with this illness. Gradually, they identified all the high-risk populations like homosexual males, promiscuous heterosexual individuals of either sex and their partners, intravenous drug users, persons who received blood and blood products prior to 1985. So this was the time I'm discussing all these events before 1985, because after that, they started screening the blood for HIV. Infants born to an already infected mother. So today the global distribution stands like this. So these are the highly infected, the brown ones. So you can see United States, this is Canada. So this is the entire US, Alaska included. That is part of US, okay, Alaska. And you have India, of course. So China is less prevalent. The disease is less prevalent in China compared to India. India and Sub-Saharan Africa, this is the Sahara Desert. So whenever you hear the term Sub-Saharan Africa, it means these. Because these are relatively richer countries, although now the political turmoil has got everywhere. So this is Egypt, Sudan, and Libya, and whatever. So these are the smaller ones like Congo and those kind of the Kenya, Tanzania, Uganda, and all those. So it is more prevalent here, slightly less prevalent in China, Russia, like that. Australia, like it's very less prevalent in Australia. The virus. So this is the virus. You know how the virus is made, composed of. You must have come across in your 12th standard. So there is a nucleic acid, the genome lies inside, and then there is an outer covering. So here we have the outermost envelope. Okay, this is known as the envelope, which has glycoproteins spiking out of it. So there are some glycoproteins embedded in it. So this transmembrane glycoprotein is known as the GP41. So glycoprotein 41. And over this transmembrane glycoprotein is another glycoprotein, which is present on the outer membrane only. That is known as glycoprotein 120. So GP41 and 120. Now inside the envelope is another layer known as the matrix. So this green layer known as the matrix, or the P17. So different proteins have been given different names. So there is P17. Inside the matrix is another layer, another covering known as the capsid. So this is the capsid. the yellow covering, and that is the P24. So this is the nucleocapsid, you can say. So there is an envelope, there is the matrix. Envelope and the matrix enclose the nucleocapsid. Nucleocapsid, that is your capsid, and then coming to the nucleic acid. So these are the nucleic acid, that is two single stranded RNAs. So it's an RNA virus, but we don't call it an RNA virus. Okay, like COVID, we call it as an RNA virus because it infects like an RNA virus. But this one does not, it goes inside like an RNA, but then it becomes a DNA. So that COVID lacks that particular step. It simply transcribes, it simply goes as an RNA and starts translating its proteins. But here it does not act in this way. It goes inside, converts itself into a DNA, integrates inside into the DNA of the host, and then produces mRNA out of that, and then produces the protein. So that intermediate DNA stage is lacking in COVID. So that those are the RNA viruses. RNA viruses include your polio virus, your hepatitis C virus, and your different other viruses. Okay, many of them. But this one is a retrovirus. So we call these as retroviruses. So there are several retroviruses, including hepatitis B and HIV and human T lymphocyte virus (HTLV). So those are retroviruses. That is, their sorry, HBV is not a retrovirus; it's a DNA virus. Forget about HBV. So we are discussing HIV now. So there are two single-stranded RNAs which function by converting themselves into DNA in the host, and then there are some proteins which help this function. So there is a reverse transcriptase, of course, known as P64. There is a protease which helps in making the. So first, the entire RNA is converted into DNA, and then it integrates into the genome. But from the proviral DNA, that is known as the provirus, right? When RNA becomes DNA and integrates into the genome, it is known as a provirus. But when that provirus gets activated, it makes a long mRNA. Doesn't make short, like, because we have individual genes and individual promoters. For them, it is just single gene and single promoter, polycistronic, right? So they make a huge mRNA. From that, it makes a huge protein chain. So that protein has to be broken down to give functional protein. So that cleavage of the protein happens through protease. P10 of the protease. There is another protein called integrase. So integrase helps in your integration of the provirus into the host DNA. So you will remember this structure. Okay. And then, so envelope and matrix covering the nucleocapsid, that is capsid, two nucleic acid, two single stranded RNAs, the proteins, that is reverse transcriptase or P64, protease or P10, integrase or P32, right? This is how it appears under the electron microscope. So now viruses are submicroscopic. You cannot observe anything under the light microscope, but inside, but through an scanning electron microscope, you can see this. These are the spikes. Okay. If you look closely, you can see the glycoprotein spikes coming out from the envelope. Okay, and then enclosing the nucleocapsid. This is a three-dimensional conformation. So that was a 2D representation. 3D, it will look like this. So these are all your glycoproteins coming out. And then if you cut open one section, you can see the matrix and the, so this is the capsid. Okay, this is the matrix, the blue. So this is the matrix of the P17. This is the capsid. Yellow is the capsid or the P24. These are the glycoprotein spikes. GP41, the transmembrane one. GP120 on the peripheral protein. It's a peripheral glycoprotein. Inside you have the two single-stranded RNAs. You have the protease, the reverse transcriptase, and the integrase. Okay. Envelope is, of course, a lipid bilayer. Envelope is basically derived from the host. Okay. The bacteria is, oh, sorry, I'm sorry, there is no bacteria here. The virus is going to, this is the virus. The RNA is the identity of the virus. That RNA is going to get converted to DNA, integrate into the host genome, make a new mRNA out of it, make new proteins, and then those proteins will combine with one more RNA that is made already, and that will make your virion. So that virion, as it escapes from the host cell, as it bursts out from the host cell, it acquires the envelope from the host cell. So if you see in the previous diagram, you can also see the MHC protein. See, so MHC, the major histocompatibility complex proteins, are also observed on the viral surface simply because it's a human immunodeficiency virus. So it has acquired the envelope from a human cell, and then that envelope will always carry the human antigen, that is MHC. So this is the genome of the virus. So now we are just looking at this, the single stranded RNA we have unfolded. So there are three structural genes: GAG, POL, and N, and six regulatory genes, that is your VIF, VPU, VPR, TAT, REV, and NEF. So these are all the, and then there is the long translated um, untranslated region. So that should be the untranslated. Okay, I forgot the acronym, the full form of it. Okay, expansion of it. But that's important for binding of the proteins to allow the transcription to happen, all those things to happen. Okay, the LTR region. So these are the structural genes. Like I told, there are three structural genes: GAG, POL, and N. So GAG is what makes your G for glycoprotein. So just remember that. No, I'm sorry. No, just forget that. Okay. So N is the envelope. Okay, N is the envelope. It is going to make your glycoprotein 41 and glycoprotein 120. So N makes up the transmembrane 41, glycoprotein 41, and glycoprotein 120. Transmembrane protein, and this is the outside protein. Why is GP120 important? GP120 is the receptor that is going to bind your CD4. So we say that HIV infects CD4 T helper cells. How it does that? Through your glycoprotein 120, which is made by N gene, structural gene. GAG structural gene is important to form both the matrix and the capsid. So P17 or the matrix, this one, the green, and P24 or the inner core layer, inner core. So outer core protein layer, inner core protein layer. So this capsid, that is P24. So P17 and P24 are made from the GAG protein. You don't need to remember the other things. POL makes the other things inside what we saw, the proteins which were present inside. P64 or the reverse transcriptase, P10 or the protease, and P32 or the integrase. Okay. So generally all these things are made as one. So GAG precursors, I think even the envelope. So GAG is made as one protein. I'm not very sure although, but just remember that GAG is made as one protein. So P17, P24 are all part of one single large polypeptide which has to be cleaved. So that protease P10 is going to cleave it. But I remember, as far as I remember, GAG and POL are all made together. But let's see. Okay, and then there are the regulatory proteins: TAT, NEV, REF, WAPU, WIF, VPU, WIF, VIF, and VPR. So they are required for your different kind of interaction. Like the virus, they allow the virus to integrate with the host genome. They prevent the host factors from attacking the virus inside the cell. So this TAT is going to strongly activate the transcription. So once the provirus has integrated, the transcription is going to be activated. REV is going to allow the export of the unspliced and the singly spliced mRNAs from the nucleus. So this we will see in the viral life cycle. So once the provirus has integrated with the human genome, hijack the genome, and starts making copies of itself. So then the unspliced mRNA, which is your genomic, which is going to become the genomic RNA, and the spliced RNAs, which are going to become your proteins, the nucleocapsid. Sorry, not the nucleo, the capsid, the matrix, and the glycoprotein envelope. So all those things. So they are exported out using the REV. NEF downregulates host cell class 1 MHC. What does that mean? See, the host cell will try to kill the virus. How it is going to kill? The host cell is going to express the class 1 MHC, more and more of class 1 MHC, which will take the peptide, one of the viral peptides along with it, and come out on the surface so that CD8 cytotoxic cell can recognize the cell and kill it. But virus is not going to allow the class 1 MHC to be produced. So it downregulates host cell class 1 MHC. It downregulates the CD4 molecule on the host cell. Okay. VBU is required for efficient viral assembly and budding. So so initially the structural genes are what the composition of the virus is, but the regulatory genes are which allows this composition to happen. So initially we saw that some gene allows the transcription to happen, other gene allows that mRNA to be exported from the nucleus. Then the third gene is going to allow for the viral assembly. Assembly means सारे प्रोटीन्स को एक साथ लाना है, P17 को, P24 को, बाहर GP120, GP41 को, और फिर budding कराना है. So that budding, so we will see in the diagram of viral life cycle, and promotes extracellular release of the virus particles. VIF promotes maturation and infectivity. Again ये भी maturation के लिए चाहिए. ये VPR शुरू के स्टेप के लिए चाहिए. Promotes nuclear localization of pre-integration complex, inhibits cell division. So after this new virus, after extracellular release is going to attack a new cell. तो वहां पे जाके वापस न्यूक्लियस में पहुंचे, प्री इंटीग्रेशन कॉम्प्लेक्स बनाए, एंड फिर वो पहुंचे. तो ठीक है, जस्ट रिमेंबर दैट देयर आर 3 structural genes and 6 regulatory genes. See, nothing of this is important. You don't have to remember these functions. ये सब सेकंड ईयर में पढ़ना आराम से डिटेल में माइक्रोबायोलॉजी में. ओके, अभी ये सब तुमको वैसे ही बता रहा हूँ. So what, like I told you, it is so powerful and it is so evil that is taking the war to the castle itself. So directly it is attacking the commander in chief. It is attacking when the hunter becomes the hunted. So it infects who? It infects the CD4 and all the cells carrying CD4 antigen. Majorly it includes the T helper CD4 T helper cells and the monocytes and macrophages. So all you can see here, the T cell mediated immune response is totally hijacked, totally taken over. That is why HIV is the king of infections, you can say. Why CD4 plus cells? Because it has GP120, which interacts with a very high affinity to CD4. But is that enough for infection? So this is a very important part, that GP120 is the receptor, but then there is a co-receptor called GP120 is not the receptor, CD4 is the receptor. Okay, in the previous slide I told this was the receptor, this was the weapon, or this was the ligand that will bind to the receptor. This is the receptor. So CD4 was the receptor, but that is not enough. So it needs another cell surface molecule, also called as co-receptor. So to which your GP41 will interact. So that is known as co-receptor. That is, on the T helper cell you have CXCR4, on the monocytes you have CCR5. So on T helper you have CXCR4, on monocytes you have CCR5. See, CXC and CC are families of chemokines. What are chemokines? Cytokines which have a chemotactic property are known as chemokines. So chemokines have 3 families. I have forgotten the third family, but one family is your CXC family, one family is your CC family. So these are CXC receptor 4, CC receptor 5, found on the T helper and the monocyte macrophage respectively. So those virus particles which use this, which attack the T helper cell and you will use CXCR4, they are called T-tropic viruses. Those which attack, the less common ones which attack the monocytes macrophages, will use CC receptor 5, CCR5, and they are called M strains. So there are T-tropic strains and there are M-tropic strains. So mode of infection, you are very well aware: infected blood, milk, semen, vaginal fluid. So unprotected sexual intercourse. The risk is higher for females because they are the passive partner. They receive a larger amount of the ejaculate. It stays for a larger time in their reproductive tract. So any ulcers or anything, we will see how it enters. Passive anal sex partners, so that is why homosexual. That is also the passive partners are more, because again the same funda, that the semen will be, the secretion will be present for a larger amount in their reproductive tract. Sharing of hypodermic needles by IV drug abusers, transplacental or breast milk transmission, transfusion of infected blood and blood products. So thalassemics and hemophiliacs who receive recurrent blood transfusion, so they are at the highest risk. Other sexually transmitted diseases, if they're present, they will enhance. So why we say that other, it is said that if a person is suffering from a sexually transmitted disease, he has a greater likelihood of catching HIV, because there is already an inflammation happening there in the reproductive tract. Inflammation का मतलब है वहाँ पे macrophages, neutrophils, lymphocytes सब present है. तो अगर already receptive cells वहाँ पे present है जो target है HIV का, HIV के लिए जो target rich zone हो गया ना. So it is like when as soon as HIV enters, it sees, oh, targets everywhere. So it's easy for those people to be infected. So anyone who is promiscuous, sexually promiscuous, needs to be extra cautious in using protection, in not indulging in reckless sexual practices. Langerhans cells are a type of dendritic cell. So like I said, the antigen antibody interaction or antigen T cell receptor interaction mostly happens in the cafeteria. क्या है? Lovers के meet होने का cafeteria क्या है? So for us, we meet our partners at the restaurants or at clubs or at movie theaters, like that, right? Or in parks, gardens, whatever. Those are our meeting places. For them, for T cell and B cell, they meet their destined partners Destined, why? Because they are pre-programmed genetically. So their marriages have been made in heaven, but they are still hunting for their partners. But where do they find their partners? Mostly they find them in the lymph nodes, in the spleen, in the mucosa-associated lymphoid tissue. These are the secondary lymphoid organs. So, and who gets their partners there? These cells, dendritic cells, macrophages. They will capture the antigen, पकड़ के उनको लेके आएंगे, lymph node पहुंचाएंगे, afferent lymphatic से आगे उनको spleen में पहुंचाएंगे. So mostly the meeting takes place in the secondary lymphoid organs. But the main people who, the agents जो हैं इनके agents बोलो इनको या जो भी बोलो, so they are the dendritic cells. So these dendritic cells with long processes reach close to the vaginal lumen, can take up the virus and then transfer this virus to the CD4. So you can see this is the dendritic cell. This is the CD4 positive T helper cell. The green particles, what you see are the HIV-1 virus. So there are two types of strains, HIV-1 and 2. But presently we are only talking about HIV-1. Mostly we'll be talking about HIV-1. HIV-2 is less prevalent and it is less infective. Many times HIV-2 HIV-2 positive individual can survive for a very long time, can lead almost a normal life. So sub-Saharan Africa you can find HIV-2 strains more commonly. But here we have HIV-1 strains. And that strain also keeps mutating. Okay. We'll see how the virus mutates or why it mutates. It may mutate to counteract the drug. If you are giving some drug, it may mutate just to change its structure, to acquire a new structure. That is what these viruses do. They mutate very fast. That is why generally we don't use specific drugs against influenza viruses and all. Common cold hota hai to tumko koi antiviral to nahi dete na. Generally you are only given treatment for cough and cold, symptomatic relief, that's all. Because antivirals are not very effective. Antibacterials, antifungals are still very effective. Their mutation is not so fast. But for viruses, the mutation happens very fast. So HIV-1 also has mutated, and those are known as clades. So different clades of the HIV virus are found. Okay. So HIV-1 dots. So entering the lion's den. So now what happens is that वो वहाँ पहुँचेगा lymph node में जहाँ ज़्यादा से ज़्यादा T cell. अगर like I said, if there are STDs, then it will find some T helper cells right there in the vagina, okay, or in the penis or whatever diffuse. But otherwise, it will reach through the dendritic cell and go through the dendritic cell and reach where any of the secondary lymphoid organs, like through the submucosa to the draining lymphatic channels through the lymph node. Here, what will happen? Here, the host will try. Of course, host sees it as a threat. Host does not know that it is going to attack itself, but then finally host has to initiate an immune response. So adaptive immune response will be generated. CD4 cell will, यहाँ पे जो meeting दिखा रहा है, तो इसमें CD4 cell is going to recognize it through an MHC peptide complex. It is going to get activated, generate effector cells, memory cells, going to stimulate the B cells. B cells will stimulate antibody production. All that will happen. But once it reaches the lion's den, reaches the lymph node, now speed of the spread is accelerated because many more cells with the proper surface receptors are encountered. Uska to receptor is CD4. CD4 is looking for it, but it does not know that it is looking for CD4 even more desperately. So it is going to just get stuck to CD4. It does not need MHC. वो MHC के साथ नहीं जुड़ेगा. Okay, MHC के साथ जब आएगा, then that is the antigen presentation. That is the proper T cell receptor antigen interaction. But this I'm talking when the virus through its GP120 rather, is going to directly attack the CD4 antigen. So you can see here the CD4, ये virus है. Through its GP120, it binds to the CD4. But I said this interaction, is it enough? No, it is not enough. So the next interaction that is required is your GP41 to interact with the co-receptor. So if assuming that this is a T helper cell, the co-receptor will be CXC receptor 4. If it was a monocyte or macrophage, assuming that it is a macrophage, the co-receptor will be CC receptor 5. So उन दोनों को वो दोनों जब चिपकते हैं एक दूसरे, उसके बाद when this gets stuck and then it migrates and this gets stuck, then it's easy for the virus to the envelope to just fuse with this. Okay, it fuses and so this is, the envelope stays outside, it merges with the host envelope, and the nucleocapsid is just driven inside the host. Then the nucleocapsid breaks open to release the single stranded RNA. Single stranded RNA is reverse transcribed first to generate an RNA-DNA hybrid. Then the RNA is cleaved using a ribonuclease H, which is also the function of reverse transcriptase itself. So that P64 also has a ribonuclease. that will cleave off the RNA and make a second DNA copy. This will become your double-stranded DNA. So that is why these viruses are retroviruses. Unlike the coronavirus, which is an RNA virus, coronavirus does not do all this. It comes inside, but it simply uses its RNA to make proteins. So it will just get translated using RNA. What is it? Using the ribosomes and all translation assembly. So all the translation assembly will come. It does not need to convert itself into DNA. Here this converts into DNA, and that DNA is now going to integrate with the. So it will enter the nucleus first, and then it will integrate. So these are the various steps: binding, and then entry, and then release of the genome and enzymes, reverse transcription, forming hybrids, then degradation of the template to give the double-stranded DNA, and formation of second copy, synthesis of second strand. So this was the first strand synthesis and second strand synthesis. Okay. Also in RT-PCR, you must have encountered this term. So in RT-PCR also you have an RNA. What we are doing for coronavirus. So coronavirus diagnosis is done through RT-PCR. That is, you have the RNA particle, you make the first copy RNA-DNA hybrid through reverse transcription, and then you make the second copy, and then you do the PCR amplification. So here also you have the first strand synthesis, forming hybrid, then breaking the RNA, then forming the second strand to form double-stranded DNA. So this viral double-stranded DNA is translocates to the nucleus and integrates into the host genome. This is known as the provirus. So formation of the provirus. So HIV first infects the target cell using all these steps and forms the provirus. Co-receptor, like I told you, co-receptor is necessary. T cell tropic strains infecting the T helper cell use CXC receptor 4. M tropic strains use CC receptor 5. So you have T cell and monocyte. T cell having the CXC4 or monocytes having the CCR5. Now these chemokine receptors are there. Which are the natural ligands? Of course, they are chemokines. Okay, natural ligands. ये तो virus के लिए तो नहीं बना था ना receptor. Receptor बना था chemokines के लिए, and chemokines are nothing but cytokines with a chemotactic property. So SDF1 is one of the chemokines that attaches to CXCR4. Or for your CCR5, it is Rantes macrophage inhibitory protein 1 alpha, macrophage inhibitory protein 1 beta. So these will just stick to it. So if they are there in plenty, they may actually interfere with they may compete with the virus, and they may virus को नहीं घुसने देंगे. ये हो सकता है. So both are receptors for chemokines. The normal ligands can block HIV infection. So that is the medical application, that individuals homozygous for a CCR5 mutation. It has been observed that some individuals who have deletion of the CCR5 gene on both the chromosomes, okay, or some mutation that renders it ineffective, they are resistant to infection. A big thing, right? So CCR5 inhibitors have also been researched into, and now they are emerging as a new class of antiretroviral drugs. So that will not allow the attachment to happen. So that can actually, it can't prevent the infection, because we'll see why preventing the infection is a problem. Because by the time you recognize the infection, it has already harbored itself in some of the cells as a provirus. Provirus बन के तो रहेगा. अब हाँ ये कर सकते हो कि new cells को infect नहीं होने दोगे, but that provirus will still become, someday it can get activated, someday it will produce infectious virion, then again तुमको drug देना पड़ेगा. So that will go on. So someday, like I said, that provirus, someday जब भी उसका मन करेगा, जब भी कुछ activation, let's say कुछ inflammation हुआ है, an activation हो रहा है, cytokines है, ये भी activate हो सकता है. Normally what will it do? It will just keep dividing along with the cell. जब भी cell division होगा, DNA multiply होगा, DNA replication होगा, the provirus will also get replicated. It will just keep on going like that as the genomic DNA. But someday it can get activated. What will happen that day? It will transcribe itself. So you can see the unspliced, ये transcription हुआ. The transcribed product has to be exported out of the nucleus. So this is the unspliced RNA, and this is the singly spliced version. So both the things, they get exported out of the nucleus. The unspliced version just stays like that. It is the genomic RNA, the single stranded RNA. Okay, it is not going to get translated. The singly spliced version will be translated first into a long polypeptide precursor, large polypeptide precursor. Then using the protease enzyme, it will be broken into its major proteins, that is your P24 or the capsid protein, P17 or the matrix proteins. And then these proteins or the other proteins, even the enzymes like P10 protease भी बनाना तो पड़ेगा ना, तो P10 protease or P64 reverse transcriptase or P32 integrase. So all these enzymes, the capsid proteins, and finally the genomic RNA will come together here to form the virion. So this is the virion or the new virus particle. And this new virus particle will, through the process of budding, acquire the envelope from the host cell membrane and become a full-fledged new virus that can go and attack a new cell. So transcription factors stimulate transcription, and then several mRNAs. The RNA is exported through the cytoplasm. Ribosomes from the host cell will be used to catalyze precursor proteins. Protease will cleave them into viral proteins. Finally, the genomic RNA and the proteins, they will assemble beneath the host cell membrane, into which your GP41 and GP120 are inserted. ये तो membrane में ही है, ये सब बनाया नहीं है अभी, ये पहले से ही बनता है. वो शुरू में when the virus entered, it left its envelope there. So then they were already present. And the membrane buds out. Release virus particle completely matured. Release viral particles, complete maturation. Incorporated virus precursor proteins are cleaved. Okay, same thing as said. So you can see this is an electron microscopic picture of the lymphocyte with the budding happening. So this is the budding. What will it do finally? It will lead to so many new virus particles will bud out from the surface of the cell that the cell will be destroyed. So there will be lysis of the cell. So normally, so there was an attempt to see how many cells are infected with HIV, but that attempt was never successful because the lifespan is so shortened. Like they destroy the cells so fast, you cannot even realize. So people thought that might not be the mode of T cell destruction, but eventually it was realized that no, they are destroying the T cells so fast that you cannot make out, because the body also is compensating by making new T cells. So the amount of prevalence of infected T cells which you can detect through tests will be less, because the infected T cells are destroyed within 1 to 1.5 days. So that is the lifespan. Okay, normally it should last for some weeks, 3 weeks, but once infected with HIV, within 1.5 days, like 1.5 days, they are destroyed. So you can see here the gut-associated lymphoid tissue. So these are the Peyer's patches. So this is the terminal ileum, and the gut-associated lymphoid tissue are known as the Peyer's patches, right? So you can see this is how it will look through the endoscope in a normal person. In a normal person, you will see a lot of lymphoid accumulations. And if you take a biopsy and stain it for your CD4 positive cells, T cells, you can see so many brown spots. The same contrasted with an HIV positive person, even in the acute stage, okay, there is complete loss of lymphoid tissue. So they attack the CD4 so badly, especially in the gut, that there will be total loss. You can see in the biopsy, very less brown stains are seen. So CD4 positive T cells are attacked, they become the provirus, they start proliferating, and they will destroy it. So latent, so why they become chronic? So this was what I was saying, that why preventing the infection or why eradicating it is difficult. Okay, why is eradication or cure not so easy to find? Because latent infection is established. That is the key for it to become chronic. So after integration, the provirus can become latent and remain unexpressed for long periods. Okay, it will just stay there as part of the host genome, not express itself. So you cannot even recognize, because all the host immune responses are directed against the virus, not against its DNA. All the drugs that we give are directed against the virus. So we'll see the various drugs. So various drugs, they are basically nothing but competitive inhibitors or non-competitive inhibitors of the enzymes like reverse transcriptase, protease, or integrase. But यहाँ पे तो अभी enzyme बना ही कहाँ है? वो तो DNA के अंदर है, right? It has not made itself into an RNA. It has not made those proteins. So your drugs are not going to really attack those latent cells. So that reservoir is still there. So integrated proviral DNA replicates in the cell division along with cell DNA. This makes the task of finding them very difficult for the immune system. Also, it establishes the reservoirs which can produce a lot of infectious viruses whenever the provirus gets activated. So that day may never come, that day may come very soon. You never know when that provirus is going to get activated. And once it does, then, I'm forgetting the numbers, but around 10 raised to 9 virions are released every day. So when it is active, then 10 raised to 9, that many. Okay, 10 raised to 9 virus particles are released every day into the circulation. When of course, that is not from one provirus, that I'm talking about a lot of provirus particles. And again, not only to the immune system, also to the drugs that we are going to give, it is difficult to find. So that is the key to HIV becoming chronic. So now once the infection has happened, what happens next? There is gradual impairment of the immune system. So it is slowly going to squeeze the neck of our immune system. So it is evil, it has reached the lion's den, now it has caught it by its neck, and it is not going to let go. But it is not going to strangulate immediately. Slowly it will just keep tightening the collar so that you will just suffocate. The immune system will just suffocate. So initially you can see the virus particle rises. So this is the acute stage of the infection, acute phase. So when the person has had an exposure, either due to unprotected intercourse or because of an infected blood product or whatever, intravenous drug abuse episode. So you get the virus, you don't realize it, of course. You know, if from history you can find out that in the past this had happened. So initially they present with some, meaning they develop some flu-like illness. They never come to the hospital most of the times. So they come only if they are aware. Otherwise they won't come. So they develop some flu-like symptoms. And this is the acute phase when the virus is very high in their body. Okay, they don't know that they are infecting others if they are still continuing those practices. So develop some flu-like illness. This is the acute phase. Then the body, of course, starts developing an immune response to it. So body, as we haven't seen the body antibody. So you can see here the antivirus. Okay, these don't go by these two things. These two are different from the antibody response. So this is the anti-HIV antibody. So here the anti-HIV antibody first appears and it stays. Okay, so anti-HIV antibodies come. Also, your T cell response picks up. So CD4 cells rise. Okay, CD4 cells against HIV will rise. So they will both counter. In fact, not the CD4 actually. I haven't shown it, but the CD8 cytotoxic cell will be more important. So these antibodies and the CD8 cytotoxic cell, they kill the viruses. If you can call it killing, viruses are not really live creatures, so they are not killed or anything. They control the replication, they control the rates, but still at a low level, the virus decreases. Okay, the virus titers will be low in the blood, but the latent infection continues. Some virus particles still there. The HIV viral load keeps low for a long time. Now this phase is variable. Okay, this is known as the asymptomatic phase. This phase is variable. This is going to last for, it can last for many time, like many years. It depends. It can last for a long period. So in this phase, there will be no symptoms as such. So this can also be called as a window period. There are two definitions of the window period. So one is the window period for a test, for an antibody test, because generally we do an antibody test to diagnose HIV. So the period before the antibody appears is known as the window period for the test. Okay, so we are mainly concerned. So generally, if you hear the term window period, it generally refers to the antibody test or the ELISA. So HIV ELISA cannot detect the presence of infection before the window period is over. So in that period, the patient will be largely asymptomatic, barring flu-like illness, but he will have very high viral titers. He can infect a lot of people. Still, he will test negative. So in the window period, you can only do PCR, polymerase chain reaction, to diagnose. So RT-PCR or PCR can detect, but not your ELISA. So that is the window period for ELISA testing. Otherwise, this is generally not called a window period. I'm sorry. Mostly it is called as the asymptomatic phase. But you can call it as window period because there is no symptoms. Okay. Okay, forget that. Just remember the window period is the duration in which your ELISA tests come negative. Still, the viral titers are very high. And this acute phase is followed by the asymptomatic phase, in which the viral loads fall low because of your CD8 response, because of your antibody response. But in this period, your tightening of the collar goes on. CD4 cells are still dropping, and finally they drop to such low levels. So this is the critical threshold, that is 200 cells per mm cube. So when the CD4 T lymphocyte count drops, so normally we have like around 1000 CD4 T cells or more than 1000. See, we have lymphocytes around 30% of the total WBC count, right? Total leukocyte count maybe, let's say 5000 or 4000. Let's call it 4000, of which 70% is neutrophil, 30% is lymphocytes. So 30% of 4000 would be around 1200. If it is 5000, around 1500. Of that, 66, 2/3 are CD4 positive, 1/3 are CD8 positive. So if you're taking 1500, 1000 will still be CD4 positive. So normally we have CD4 positive lymphocytes 1000 per mm cube. When it drops to the critical level of 200 per mm cube, then the person goes into the third and terminal phase of the disease known as AIDS, acquired immunodeficiency syndrome. So what happens in this? Because the T cell counts have dropped so low, now you will have all that spectrum which we saw in the initial slide. What happens when T cell defects are there? Which opportunistic infections develop? So opportunistic infections like viral diseases, fungal diseases like candidal ulcers, or protozoal diseases like pneumocystis carinii pneumonia, even malignancies. So because immunosurveillance of malignancies is a function of which cell? CD4, CD8. So CD8 cells generally destroy the infected viral cell, virus infected host cells, or they destroy the cancer cells. You remember the two antigen processing pathways? One was the cytosolic pathway and one was the endosomal or the endocytic pathway, right? So that endocytic pathway is used for extracellular pathogens. For intracellular pathogens like viral pathogens, like cancer pathogens, you have the cytosolic pathway, which presents through MHC1 to CD8. So all that also is getting affected. So you will have cancers like Kaposi sarcoma. So all that starts developing here. And here, now your viral titers are going to go through the roof. Virus will keep going high. Finally, even your antibody responses will give way, because once the T helper cells are destroyed, your B cells also cannot function properly. So everything will go. Finally, the host will be left totally susceptible to everything, and he will die. Most commonly, the death is because of tuberculosis or severe diarrheal disease or like that. So around 9 to 11 years after acquiring the infection, if there is no treatment, the person will die. So this is the normal life cycle. So that was the viral life cycle in the cell, okay, the previous diagram that you saw. And this is the phase of the HIV illness, phases of HIV illness. So stages of HIV illness: acute or primary stage, when there is only flu-like illness, there are no HIV antibodies, there is a very high viral load. Asymptomatic period or window period. So, okay, so this also is one definition of window period, but mostly when we refer to the window period, we are talking of the HIV ELISA window period. So window and asymptom, because there are no symptoms basically. So antibody and cytotoxic T lymphocytes, they check viral replication. So viral load goes low, but it does not stop. So it is continually multiplying, and there is gradual decline in CD4 T cells. AIDS is the final stage, when the CD4 T cells gradually declining reach a threshold limit of 200 per microliter. So delayed type hypersensitivity reactions are lost. A lot of opportunistic infections are there. So AIDS, severe decrease, it's a terminal stage of HIV illness in humans. Severe decrease in T cells is the clinical hallmark of AIDS. There is direct viral infection and destruction of the CD4 T cells, obviously, yes. See, direct viral infection induced death is known as the cytopathic effect of the virus. What do I tell you? So the virus, see, HIV causes illnesses in two ways. One, it is going to impact your immune system, weaken it. So it is going to cause an indirect illness. Indirectly you will be susceptible to tuberculosis, to candidal ulcers, to pneumocystis carinii, to malignancies. One, it also causes direct death. So direct death is seen in case of CD4 T cells. So it is not seen in candidal ulcer. The mucosal injury of your is being caused by candida. Okay, candida albicans. Similarly, or tuberculosis is being caused by mycobacterium tuberculosis of it infecting the lungs. It is not by HIV directly. But here, this is HIV directly. Similarly, in brain also, HIV directly infects the cells and kills them. So some of the activities are direct cytopathic effect. It is called CPE, or cytopathic effect of the virus. Whereas the others are mostly indirect because of immunodeficiency. So direct destruction of CD4 T cells. Smaller numbers of CD4 T cells may remain infected without actively replicating. So these are your latently infected cells. Absent delayed type hypersensitivity reaction to which the individual was normally reacting. Decreased serum immunoglobulins. So eventually